glycation is a process that is inevitable. So it means it happens in all cells that and all organisms that use sugars and glucose for producing energy. And we know this process is also age associated. So as we age, our proteins and nucleic acids will get modified, will get sugars added to them. So this is what glycation is all about, is the addition of sugars to molecules like proteins and nucleic acids. And what this does is that this somehow damages proteins and nucleic acids and causes them to stop functioning properly and eventually to change their structure and maybe accumulate and cause problems to the cells and eventually to the organisms where they deposit. This work on glycation started already several years ago while I was still in Lisbon, in Portugal. So one of my students at the time came to the lab. He was working with other colleagues in Lisbon that were specialists in studying glycation. And we had this idea that maybe glycation could also affect alpha-synuclein, because we know alpha-synuclein is accumulating in the brains of patients with Parkinson's disease, dementia with Lewy bodies, multiple system atrophy, and other diseases. So we thought that because it's an age-associated modification, perhaps it could damage alpha-synuclein and cause problems. And this is how we started. We started looking at this, and we first found that surprisingly, glycation was not necessarily causing the formation of the Lewy bodies that are typical in the brains of Parkinson's disease, or these types of aggregates like Lewy bodies that we can study in model organisms. So what it was doing was promoting the accumulation of other species that we call oligomeric species that we think are actually the culprits. We think these are more toxic than the Lewy bodies. And then we did a series of studies in different model organisms, in mice, in Drosophila, in collaboration with other colleagues, and we found that indeed animals that express more alpha-synuclein in a situation that mimics certain genetic forms of Parkinson's disease or aging, glycation could cause the protein to accumulate and to cause toxicity in certain brain areas, and that this was actually also associated with behavior alterations in simple models like a fruit fly that we use routinely as a model in the lab. And now more recently, we've been looking at other aspects of glycation, not just by agents that we think are stronger glycating agents, but other sugars, because we think glycation is perhaps the missing link or one of the missing links between diabetes and Parkinson's disease. We know that patients with diabetes have an increased risk for developing Parkinson's, but we don't know the molecular mechanisms that explain this connection. So we think that glycation may be one of these connecting factors that could damage proteins, cause them to accumulate, and eventually lead to disease.